set.
They get away first time. Tyson Gay right alongside Usain Bolt, but here he goes, streaking away already. It's Bolt all the way. He's looking round at Gay. Watch the clock. It's goal for Bolt, and again he's done it again. A new world record for Usain Bolt. They say lightning doesn't strike twice. Can you believe it? He is flying. The world belongs to Bolt. Berlin belongs to Bolt. 9.58. Stunning, absolutely. They are adjectives which are inadequate to describe this man.
That was Usain Bolt setting a phenomenal world record. It may seem a little straightforward, putting one foot in front of the other, but the physiological processes involved in stimulating a muscle contraction are actually incredibly complex and become mind-boggling when you think about how rapidly they occur in this situation. As mind-boggling as it might be, we're going to be breaking down this process in the present session, where we analyze excitation-contraction coupling and the sliding filament theory. Excitation-contraction coupling describes the process by which an action potential passes across the muscle cell membrane to stimulate contraction, and the sliding filament theory describes the process by which the thick and thin filaments, which we discussed in the previous session, actually produce a muscle contraction. Together, these two connected processes describe the process by which a skeletal muscle contracts in response to electrical stimulation. The objectives for this session: first, we'll look at the process by which an electrical stimulus produces muscle contraction in relation to the structures discussed in the previous session. Next, we'll look at some principles behind muscle contraction and discuss the concepts of twitch, tetanus, force summation, and the length-tension relationship. We'll then dive into the concept of muscle fiber types and finish off with a discussion of muscle fiber degeneration and regeneration. We'll start with the process of excitation-contraction coupling. This is the process by which the electrical stimulus from an alpha motor neuron reaches the thick and thin filaments to produce a muscle contraction. Now, it's important to remember how incredibly quick this process takes place. Sitting and listening to this video right now, you're probably taking notes, either through pen to paper or a keyboard. Whatever method you use, think about the rapid and precise muscle contractions you are using to accomplish this task. It really is incredible when you think of all the steps involved. For this rapid process to take place, an electrical stimulus from the alpha motor neuron is converted into a chemical release of neurotransmitter, which then triggers an electrical stimulus across the muscle membrane. That, in turn, triggers a chemical release of calcium from the sarcoplasmic reticulum, resulting in a muscle contraction. Okay, so let's back up for a moment and take it from the beginning. The process of muscle contraction is initiated when an action potential reaches the axon terminus of an alpha motor neuron, triggering the release of acetylcholine into the neuromuscular junction. That process of acetylcholine release is a topic for another course, so we won't go into those specifics here. We pick things up with the binding of acetylcholine to the acetylcholine receptors in the gap junction membrane. This binding opens a voltage-gated sodium channel within the receptor, and sodium is able to rush into the sarcoplasm, triggering a localized action potential within the sarcolemma inside the neuromuscular junction. This localized membrane depolarization is sensed by sodium channels along the periphery of the neuromuscular junction, causing them to open and allowing further sodium into the cell. This, in turn, triggers sodium channels even further out to open, and the membrane depolarization spreads away from the neuromuscular junction in all directions as an action potential. Here's a way to think about the spread of an action potential. You're familiar with the concept of a wave at a sporting event, right? Sure you are. And if you've ever been in the stands for one, you know how it works. When you see it start up, you wait for the person beside you to stand up before standing up yourself a fraction of a second later. Now, imagine for a moment that each one of these people represented a voltage-gated sodium channel, and that instead of standing up, the opening of an adjacent sodium channel caused neighboring sodium channels to open a fraction of a second later. Similar to what we see with the movement of a wave through a crowd, you get the impression of what an action potential spreading across the sarcolemma would look like. So that covers the spread of an action potential across the cell membrane. But remember, we also have this T-tubule network that projects into the internal environment of the cell. Seeing as this is a continuation of the sarcolemma, you'd expect that the action potential would follow the T-tubules down into the depths of the muscle fiber. And that is precisely what happens. Again, if I were to make an analogy here, well, this is where my mind goes to.
The target area is only 2 meters wide. It's a small thermal exhaust port right below the main port. The shaft leads directly to the reactor system. A precise hit will start a chain reaction which should destroy the station. Only a precise hit will set up a chain reaction.
You're all clear, kid. Now let's blow this thing and go home.
Yeah, I know, major Star Wars geek. This allows the action potential that started on the surface to pass in and around all the myofibrils, bringing the electrical potential to the contractile apparatus. All right, so this gets the action potential into the depths of the muscle fiber, but how does that translate into muscle contraction? You'll remember from our last session, we talked about the relationship of the terminal cisternae of the sarcoplasmic reticulum to the T-tubules, forming this triad structure. The passing of this action potential through the T-tubule system triggers calcium channels in the sarcoplasmic reticulum to release calcium into the sarcoplasm, flooding the myofibrils with this ion. We'll look at what happens when this occurs in a short while, but first, let's take a closer look at this interaction between the T-tubules and terminal cisternae. This is a schematic close-up view of a centralized T-tubule surrounded by 2 terminal cisternae. In addition to the sodium channels that help to propagate the action potential, T-tubules are also rich in a tetrad of L-type calcium channels. The channel was originally referred to as a dihydropyridine receptor, as it was originally discovered as the target for this class of antihypertensive medications. While the name is somewhat archaic, it still appears widely in the literature, including in Step 1 prep materials, so we'll continue using it here. In the sarcoplasmic reticulum membrane, we find another calcium release channel, the ryanodine receptor. Again, a bit of an archaic name. In this case, it's because of the channel sensitivity to the plant alkaloid of the same name. So here's what seems to be happening. There appears to be a physical coupling between the dihydropyridine and ryanodine receptors. When an action potential passes along the T-tubules, the dihydropyridine receptors are open, allowing a small amount of calcium to enter the cytosol, that is the sarcoplasm, within the muscle cell. This doesn't have much of an effect on the myofibrils themselves, but it does trigger the opening of the ryanodine receptor channels, which allows massive amounts of calcium to flood the sarcoplasm. This is the source of calcium that triggers muscle contraction. We'll move into a discussion of the sliding filament theory in just a moment, but before we do, a couple of quick notes on these 2 calcium channels. First, dihydropyridine. As previously mentioned, this channel was originally named because of the influence of a class of drugs known as the dihydropyridines, which serve as antagonists, blocking the calcium channel from opening and from triggering depolarization. This class of drugs were used as antihypertensive agents and include amlodipine and clofazimine. Note, however, that their effect is on vascular smooth muscle rather than skeletal muscle. Another class, known as the non-dihydropyridines, act on cardiac muscle. Now, the ryanodine receptors. Like many proteins found within the body, there are different isoforms of the ryanodine receptor, and one in particular can have deadly consequences during surgery. Some inhaled anesthetics can trigger this isoform to remain open, resulting in widespread muscle spasm and, as a result, hyperthermia from the heat generated from the muscle contraction. The condition is called malignant hypothermia, and although rare, it can be life-threatening. Early recognition of this condition is critical for successful treatment, which involves administration of dantrolene, a ryanodine receptor antagonist. Okay, time to shift gears and look at the sliding filament theory of muscle contraction, involving the thin and thick filaments discussed in the previous section. In resting skeletal muscle, the thick and thin filaments are uncoupled due to the presence of tropomyosin blocking the myosin binding site on the actin monomers. Each myosin head contains a single molecule of ATP, or more accurately, a hydrolyzed molecule with both the ADP and inorganic phosphate molecule remaining bound to the head. This is considered an energized state, with the myosin head in a cocked position, primed and directed vertically, and ready to engage the binding site on actin once exposed. With excitation-contraction coupling, calcium floods the myofibrils, having been released from the ryanodine receptor calcium channels. This allows calcium to reversibly bind to the troponin C discussed in the previous session, which leads to tropomyosin shifting its position, exposing the actin binding site. With the myosin binding site now visible on the actin molecule, the myosin head is able to reach up and bind to the myosin binding site, forming the transient actin-myosin crossbridge. Binding of the myosin heads leads to dissociation of the inorganic phosphates from the heads of the myosin molecule. The release of the inorganic phosphate molecule provides the energy for what is known as the power stroke. This involves a conformational shift in the myosin molecule at the hinge region, returning it to what can be called the uncocked position at 45 degrees to the long axis. The swivel moves the myosin head a horizontal distance of 11 nanometers, which is equivalent to 2 actin monomer units. As the myosin is physically bound to actin while this is happening, the actin filament also moves an equivalent of 2 monomers in space. As a result, each power stroke causes the thin filament to shift position relative to the thick filament. So this is where the term sliding filament theory comes from. After the power stroke is complete, the myosin head must detach from the thin filament. This requires a binding of a new molecule of ATP to the binding site, displacing the old molecule of ADP. Okay, so if a new molecule of ATP is needed to break the crossbridge, does that mean that if there is no ATP, the myosin head will remain bound indefinitely? Well, yes it does. But ATP levels are highly stable in muscle cells, even under the most intense activities. Except, of course, after death. In the first few hours following death, the sarcoplasmic reticulum deteriorates, releasing calcium into the muscle, triggering muscle contraction. As there is no oxygen to supply aerobic metabolism, ATP stores are quickly depleted, and the myosin head remains attached in what is known as a rigor complex. This is the condition known as rigor mortis, which peaks around 12 hours after death. Over the next few days, myofibrils are gradually deteriorated through the actions of proteolytic enzymes released from the lysosomes, and the muscle is once again pliable. Okay, can't follow the crossbridge cycle any further in deceased muscle, because it leads to a dead end. Pun definitely intended. Let's return to living tissue to complete the cycle. Immediately after uncoupling from the actin molecule, the myosin head hydrolyzes the ATP molecule once again into ADP and inorganic phosphate. And as we mentioned before, both molecules remain bound to the myosin head. This hydrolysis and the energized state of the myosin head causes a swivel at the hinge region, returning the head to the cocked, primed position. If calcium is still present in the sarcoplasm, the tropomyosin will still be swiveled away from its resting position, and the binding site will still be exposed. In this case, the myosin head will immediately re-engage the actin filament, causing another power stroke that will further move the actin molecule another nanometers in space, and the cycle will continue to repeat. It's also important to note that while we are discussing a single actin-myosin crossbridge complex in this example, there are thousands of crossbridges that are working asynchronously together to generate this contraction. The entire process is not that dissimilar to what we see in rowing, where an oar is dipped into the water and a powerful stroke propels the boat along through the water. If we think of the boat as being the thick filament, the ores as the myosin heads, and the water itself as the thin filament, we get an appreciation of how this cross-bridge cycling can slide the thin filament along relative to the thick filament. Only difference is that in rowing we have synchronicity, with all the oars moving in phase with each other. In muscle, the thousands of cross bridges can be in any phase of the cycle at any given moment in time. This asynchrony results in more fluid contractions rather than bursts of force. Okay, so we discussed the process by which actin and myosin slide across one another, but what does that mean at the level of the sarcomere? It's important to keep in mind that the left and right side of each sarcomere represent a mirror image to each other, such that the cross bridges on either side work to pull the thin filament closer to the center. As a result, we notice that the two regions where no overlap is observed, in this case the H zone in the middle and the I bands out to either side, those will shrink in size. On the other hand, the full length of the A band represents the distance across the entire thick filament. As the thick filament itself does not shrink in size, we don't see any change in the length of the A band during contraction. So as we said, this process will continue indefinitely so long as calcium, and as we've seen from rigor mortis, ATP, remains in the myofibril. When the stimulus is removed from the muscle, the ryanodine receptors close and calcium levels in the myofibrils drop. When this happens, the troponin-tropomyosin complex returns to its resting state, blocking the myosin binding sites on the thin filaments. The myosin heads, energized and in the cocked position, lay waiting for the next stimulus to arrive. The removal of calcium from the myofibrils is not a passive process. The sarcoplasmic reticulum membrane is also rich in a second protein, the sarcoplasmic endoplasmic reticulum calcium ATPase, which is a bit of a mouthful, so let's just call it the SERCA pump. This protein is constantly active, mopping up calcium from the myofibrils. Contractions are therefore dependent on the opened ryanodine receptor in the active state to provide a constant influx of calcium. Another protein found within the sarcoplasmic reticulum is calsequestrin. There is an absolutely massive concentration gradient that the SERCA pumps are working against, with the internal environment of the sarcoplasmic reticulum accumulating 10,000 times the concentration of calcium within the sarcoplasm. Calsequestrin binds free calcium within the sarcoplasmic reticulum, which helps to diminish this free calcium concentration gradient. It's also localized to the terminal cisternae within the triad complex, meaning that there are higher concentrations of calcium close to the ryanodine receptors, where they are ultimately released from. The result is a cycling of calcium being released from the terminal cisternae and being taken back up along the rest of the length of the sarcoplasmic reticulum. Okay, it's time to step back out of the muscle fiber and look at the big picture of the muscle as a whole. The first thing to discuss is the concept of the motor unit. For any given muscle, we have a variable number of alpha motor neurons entering the cell to innervate muscle fibers, but it's not a one-to-one basis. Close to the end of the axon, these alpha motor neurons branch, with each branch going to a different muscle fiber. A motor unit is made up of a single alpha motor neuron and all of the muscle fibers it innervates. For muscles requiring precision control, such as the ocular muscles controlling the eye, the ratio is pretty low, with a single alpha motor neuron innervating as few as 5 muscle fibers. For larger muscles that are often required to generate high amounts of force, this ratio can increase to a single alpha motor neuron supplying up to 1,000 fibers. The important thing to remember is that when a specific alpha motor neuron is triggered, all of the muscle fibers it supplies will contract in unison. This is known as the all or none principle. It's like turning the lights on in a large lecture hall. You may have 5 separate switches that each control a set number of lights. Assuming that all of the fluorescent bulbs are working, when one of the switches is pressed, all of the lights connected to that particular circuit will light up, while any bulb on a separate switch will remain dark until such a time as its own switch is activated. So we've established that a muscle will respond to an electrical stimulus by shortening, and the source of the electrical stimulus doesn't particularly matter. It can be from the motor cortex or the central nervous system, or from an external source. If a single stimulus is received by a muscle, there will be a period of force development as the myofibrils contract that can last from anywhere from 10 to 100 milliseconds. This is followed by a period of relaxation lasting about the same length of time. This increase and decrease in tension can be measured across the joint that the muscle is acting upon and is referred to as a muscle twitch. If a second stimulus is given after the muscle returns to a state of full relaxation, then we will see a second twitch of essentially equal time and peak force. On the other hand, if the second stimulus is given before the muscle has had an opportunity to fully relax, it will be able to piggyback on top of the first, and the two twitches will combine to generate a greater amount of force. Keep adding more and more stimuli, and the effect continues until a plateau is reached, called a tetanus. In this third image, we see an unfused tetanus. This is because of fluctuations in force generation due to the slight relaxation that occurs between each stimulus. But if we were to introduce the stimuli so rapidly that the muscle does not have time to relax, we more or less have a smooth force line of maximal force generation. This concept is known as temporal summation, as it reflects the timing that the stimuli are given. Generally speaking, the central nervous system typically fires at a sufficient rate to always ensure a fused tetanus and smooth muscle contraction. So here's the question. If the brain is always shooting for 100% temporal summation, how can we adjust the strength of a given contraction? I mean, there are times when we want to use all of our strength to accomplish a task, and times where this would be overkill. It turns out there's a second type of summation that we have far greater control over. This is called spatial summation, and it involves the total number of motor units that are recruited to accomplish a contraction. Keep in mind that we don't have to activate all of the motor units in a given muscle for every contraction. With spatial summation, we selectively recruit the total number of motor units needed to accomplish a given task, based on motor planning and proprioceptive feedback. Normally, the body will recruit the smallest motor units first, then progressively add larger motor units when greater amounts of force are required. This is known as the size principle of motor unit recruitment. The activated units are also not static. If a submaximal contraction is held for a prolonged period of time, the motor units first activated will ultimately fatigue. When this happens, the body will switch to a different group of muscle fibers, allowing those that were originally recruited to recover. Another principle to consider is the length-tension relationship. There is a concept in weightlifting known as the sticking point, which is an optimal muscle length for generating force. Go outside of this range, and you lose the ability to generate contractile force. It's related to filament overlap in the sarcomere. The concept is pretty straightforward. The better the overlap of thin and thick filaments, the more cross bridges that can be formed and the more force that can be generated. At longer lengths, the I band and H zones increase in size, indicating less overlap between the thin and the thick filaments. This results in a progressive loss in the ability to generate force as the muscle continues to lengthen. At the other end of the spectrum, the muscle can shorten to the point that the actin filaments pass through the M line and overlap one another, and the thick filaments butt up against the Z lines. This bunching up also leads to deficits in force development. In addition to the force generated by the contractile component of the muscle, there are also elastic components within the connective tissue layers that also contribute to force development. The elements play an important role at longer muscle lengths when the contractile component fails and prevents the muscle from tearing. We can also take advantage of this elastic component during certain activities. Think of a baseball pitcher or football quarterback. There's a wind-up phase during these throwing activities in which the torso twists first away, then towards the direction of the throw. This is designed to stretch the elastic component in the shoulder and elbow flexors so that the recoil force can be added to the contractile force making for a more powerful contraction. Try throwing a ball without this windup and see how far it'll travel. A few minutes ago, we were discussing the concept of muscle twitches, and I mentioned that a twitch could last anywhere from 10 to 100 milliseconds. Heck of a range, isn't it? I mean, can we narrow it down a little? Well, no, we can't. The reason is that there is variability in the rate of force development based on the mechanical properties of the muscle. It turns out that there are different isoforms for both the myosin light chain and heavy chain proteins. The three most common types of myosin heavy chain isoforms are classified as type 1, type 2A, and type 2X, which in some of the literature out there is called type 2B. Generally speaking, a given muscle fiber will only express one of these three different isoforms. This is an immunohistochemical stain that stains myosin heavy chain 2A dark, 2X lighter, and 1 not at all. Although the stain is specific for myosin heavy chain isoforms, we also find that certain isoforms of myosin light chain proteins, as well as metabolic enzymes, are closely associated with the isoform of myosin heavy chain being expressed. This has led to the characterization of fibers as being type 1, type 2A, and type 2X or 2B, depending on the source you're looking at. From a physiological standpoint, the principal difference between these different isoforms is with the enzymatic rate of ATP hydrolysis within the globular head. The type 1 isoform has a slow rate of force development and relaxation. Fibers expressing type 1 myosin heavy chain protein are commonly referred to as slow oxidative or slow twitch fibers. In addition to MHC1 expression, they are adapted for oxidative metabolism, with plenty of mitochondria and myoglobin, which helps bind oxygen and retain it in the cell. Myoglobin is what gives dark meat and fowl its characteristic appearance. These muscles are also entirely composed of slow twitch fibers. We see a lot of these types of fibers in postural muscles that tend to be chronically activated when standing or moving around. Type 2A fibers that contain a preponderance of myosin heavy chain 2A, which has a faster rate of force development and relaxation. This allows for faster force development and more powerful contractions. But because of the faster rate of relaxation, they require a higher frequency electrical stimulus to reach tetanus and cycle through ATP at a faster rate. These are known as the fast oxidative or fast fatigue resistant fibers. Now, despite this name, they don't have quite as much mitochondria as slow oxidative fibers, and they cycle through ATP at a faster rate and therefore fatigue much faster. We see a higher concentration of these types of fibers in muscles that are used transiently and for shorter durations, such as many of the muscles of the upper limb. Last are the type 2X or 2B fibers that contain myosin heavy chain 2X. These have the fastest rates of force development and relaxation, and the fibers themselves have very little mitochondria or myoglobin. These represent the white meat muscle we see in poultry. These fibers are not particularly common in humans. Their presence normally suggests a fairly sedentary lifestyle, as they are found in greatest proportions in muscles that are used very infrequently. Now, a moment ago I talked about the concept of white meat and dark meat in poultry, and how each of these muscles contains either fast or slow twitch muscle fibers. Not the case in humans. As we saw in that slide of human muscle, all three types typically coexist in the same muscle cell. Instead, fiber type appears to be regulated at the level of the motor unit. All fibers within a given motor unit tend to be of a particular type. It's also important to note that we typically don't see fiber groupings. The fibers within a single motor unit are typically spread out over a wide area and interspersed with the fibers of other motor units. As a result, when we stain for a particular type of fiber, we tend to see a checkerboard appearance in the muscle, rather than clumping of fiber types together. This brings us to an important physiological question, and one of my biggest pet peeves, to be honest. Does fiber type conversion exist? In other words, can a type 1 muscle fiber suddenly become a type 2A or 2X fiber and vice versa? The answer is an emphatic yes, and I appear to be living proof. Here are 2 micrographs of muscle tissue taken from my own right vastus lateralis muscle. The one on the right was taken just prior to going in for an ACL reconstruction surgery. I was actually training pretty intensively in preparation for doing this study. The image on the right was taken 6 weeks after the surgery. I was weight-bearing by that point, but had gone through a long period of inactivity. Notice a lower population of type 1 fibers post-surgery. Still, despite this work, there are still people out there who teach that fiber types are set in stone, and it drives me crazy, to be honest. If my own muscle isn't proof enough, here's a definitive research article from 1991 in which biopsy samples were taken from paraplegic patients in the months following a spinal cord lesion. As demonstrated with my own study, we find time and time again that the neural input and weight bearing are critical for maintaining the type 1 fiber population. Once the muscle fiber is no longer experiencing neural input or force generation, we see a pretty dramatic conversion to the type 2 fiber types. Last topic we need to touch upon is muscle damage, as it will come up during our discussions of various muscle diseases. One of the biggest culprits in propagating muscle damage is also the molecule so critical to contraction, calcium. In muscle damage, we typically see damage to the sarcoplasmic reticulum and leakage of calcium into the sarcoplasm. This can be the result of mechanical trauma, such as a crush injury, or micro tears within individual fibers from overly forceful contractions. Normally, we would expect elevated calcium levels to result in chronic contraction and muscle spasm. With mechanical trauma, the myofibrils may also be damaged to a point that contractions are no longer possible. Or if only a small number of fibers are affected, the fiber contractions are imperceptible at the level of the whole muscle. This chronic elevation in calcium will ultimately trigger the activation of proteases, which will start breaking down the contractile apparatus. At the same time, phospholipase enzymes are also activated, which results in the breakdown of intracellular membrane components and the development of free radicals, which cause further damage. The end result is a necrotic zone at the site of damage. On either side of the necrotic zone, the healthy portions of the fiber retract away from this damaged region, and the intracellular membranes collapse between these regions to effectively create a barrier protecting the undamaged regions from the effects of the elevated intracellular calcium in the damaged regions. In the necrotic zone, this initial damage is followed by infiltration by inflammatory mediators, including macrophages to clean out debris, and the complement cascade to facilitate cytokine production. We already discussed the process by which satellite cells will proliferate and align in the center of the damaged area, forming a new myotube. So we won't go into the details of that here, except to say that as the myotube expands, the membranes will ultimately fuse with the closed-off portion of the undamaged ends of the fiber. As a result, you would have an immature myotube forming in between regions of healthy, undamaged muscle tissue.
What does it all mean?
That's going to do it for our discussion of normal muscle histology and physiology. In this present session, we discussed how an action potential is generated at the motor end plate through the opening of acetylcholine receptors, travels along the sarcolemma in a wave-like pattern, and into the depths of the muscle through the T-tubule network. This triggers dihydropyridine receptors in the T-tubule network to open the ryanodine receptors in the sarcoplasm reticulum to release calcium into the sarcoplasm, flooding the myofibrils. Calcium binds troponin C, causing a conformational shift in the tropomyosin that exposes the myosin binding site on the thin filaments. This allows the primed myosin head to bind, forming the actin-myosin cross-bridge complex. Release of the phosphate molecule triggers the power stroke, shifting the actin molecule relative to the myosin. Another molecule of ATP will displace the ADP molecule, which causes the myosin head to detach, triggering the breakdown of ATP into ADP and inorganic phosphate, priming the head for the next round. Once the stimulus is removed and the calcium is soaked up by the circa and stored by calsequestrin, the muscle can now relax. We also talked about what makes up a motor unit, what a twitch and tetanus are, and the difference between spatial and temporal summation. Finally, we discussed the properties and distribution of type 1, 2A, and 2X, sometimes known as 2B, muscle fibers, and finished off with a discussion of the mechanisms of muscle damage. Hopefully you found this session useful in preparing for our in-class discussions of muscular diseases. Until that time, this has been Dr. Stuart Inglis. Enjoy the rest of your day.